Okay, back for second video. We are going to go through and talk a little bit more about Mendel versus non-Mendelian genetics. Again, kind of going through talking linked genes, just things that you probably want to expand on or actually go through and just maybe review a little bit more before we get to the exam. I think the difference why we kind of talk Mendel versus non-Mendelian, because Mendel was a big component, obviously, to being the father of genetics was a big part of the genetic world. And so what he showed us, there was nothing wrong with what he showed us and what he went through and actually, and what he recorded, I guess. But again, there's a lot more that goes to it than what he basically showed us. So, I think it's a big part that we talk about the differences, that not everything he did show us was necessarily, right in the case of that there's so much more than just what Mendel showed us. So anyway, kind of giving credit to Gregor Mendel, there's 4 different conclusions that he come up with when he went through and was studying the pea plants many years ago. Number one, multiple alleles for the same trait. So when you guys go through, and again, we take a look at the chromosomes, again, we're going to have multiple alleles that are going to be found on those chromosomes. For a specific example would be the flower color that he studied, and that there is a flower color for allele for the white flower color, and there's allele for the purple flower color. No different than us going through and taking a look at polydactyl. They've got alleles for 6 fingers, they got alleles for 5 fingers. And so every trait that we look at, there's going to be multiple alleles. 2, and organisms inherit 2 alleles for each of those traits. You get one from mom, one from dad. I think that's pretty easy to understand. Going back to meiosis, we start looking at, again, the whole production of the gametes, the sex cells, and the whole reduction division of the diploid to haploid cell, where we want to basically separate the genetics into, again, one allele of each of those traits. So that way, when the sperm and egg come together, you're going to get that individual that has the 2 alleles for those traits. So that's true. We've also found that in the past, what researchers have found is that some of our traits actually are not just about 2 alleles, that there can be more than 2 alleles that will actually develop a trait. Dominant allele is fully expressed. Recessive allele has no noticeable effect. Again, going through, if we take a look at a dominant allele, it being fully expressed, we kind of look at the genetic side of this. We look at the more cellular side of this, is that the whole production of a protein, and that a dominant allele is generally going to be all about the production of a protein, where the recessive alleles are generally lack the building of a protein, and that's generally what gives us that recessive condition if you happen to have one or more of those alleles, right? Or I shouldn't say one or more, obviously 2 or more of those alleles, but again, we kind of look at this on different levels, in that in the case of some of our dominants, like incomplete dominants, where you can have the recessive allele and dominant allele that come together, and they're going to give you more of an intermediate form. So it kind of reduces the expression of that dominant trait. And then we take a look at 4 2 alleles of the trait segregated independently of each other. Again, as you go through and take a look at the whole production of meiosis, again, 2 alleles. As meiosis is occurring, they're going to separate from each other, meaning that basically there is an equal production or equal chance that each of those can be given to the offspring. So again, kind of let's just again that random choice, that random chance that you could get the purple flower, the white allele, and so on down the line. So Gregor Mendel, again, had a lot to do with the individuals that we see today, and the genetics that they have been able to go through and find. But we also understand that as we go through and take a look at our Punnett squares, as far as dealing with Mendel, is that there's a lot of, dominant versus recessive, which is true for a lot of our traits, but there's much more going on in the case we start looking at the whole non-Mendelian genetics. So when we start looking at non-Mendelian genetics, now we start looking at all the different types of crosses that can occur with, I guess, that kind of expanded on what he showed us. And the simple fact that we start looking at it's not always just dominant versus recessive, and that we can actually have what they call incomplete dominants. We can have codominant types, to where incomplete dominants, again, you find that intermediate form. The codominants, you find, where both alleles are expressed at the same time. So a lot of different things can happen there. Probably be able to provide some examples as we go on. And again, this can be different for levels of organization. And so as you kind of start looking at, the cellular level versus an organismal level, again, those can change a little bit, as we've talked a little bit in the past with sickle cell anemia. And that sickle cell anemia can actually thought to be codominant because both alleles are expressed, but it's an intermediate form when you start looking at the overall effects of the human body. Some of the other ones we talk about, multiple alleles, ABO blood type. It's not just two alleles. You've got three alleles that go on. Pleiotropic or pleiotrophy affects more than one character. So sickle cell anemia, again, doesn't just affect the shape of the cells, but can affect so much of the body because of basically the shape of the sickle cells itself. We've got epistasis. We've got basically one gene will turn on another. You got polygenic inheritance, albinism, where you've got basically many genes are incorporated into that. So there's a lot of different situations that are occurring during that. Non-disjunction disorders, you got deletions that can occur. You've got replications, inversions, you got translocations of the DNA, and therefore that can lead to some of these genetic disorders or gene alterations that are happening. So a lot of different things that can happen. Now, we've also talked in the past about sex-linked traits or chromosomes, that not all of these are going to happen on the autosomal chromosomes, which in our body would be the first 22. We start looking at even those that happen to the X or Y. In that, again, these are going to be sex-linked, because generally if you find, not a lot of these are going to happen on the Y chromosome, but if they happen on the X, we start seeing these more of a kind of male pattern trend. In that, again, when you start looking at, well, the men have just the one X, so what they get basically is what they get. The females have the XX, and so therefore they kind of go back to the original rules that we followed in the past, where if dominant versus recessive, they've got to have it on both of the X's in order to actually establish these conditions. So again, hemophilia, male pattern baldness, red green colorblind kind of fall into that. Now, the other part that we start looking at is when we start looking at Mendel versus non-Mendelian traits, is this whole part of these Punnett squares. And a lot of times these Punnett squares hold true if genes or if chromosomes are found on if alleles are found on separate chromosomes. Now, they've also done some studies in the past where they've found that alleles that are close together due to crossing over can actually cross over together, which basically means that they are linked. And how they determine this, and I'm going to scroll all the way down to the bottom here, is we start looking at the number of recombinant individuals, which are the number of individuals that are different from the actual parents themselves. And so if we take a look at a lot of these traits, a lot of these conditions that we pass down from parent to offspring, is that, a Punnett square like this should end up as a 25, 25, 25, 25. But when you take a look at it, it obviously doesn't end up as a 25, 25, 25, 25, which shows that something is occurring there, and that basically you're starting to look at some of these genes are linked because there's a very small amount of recombinant individuals compared to the parents. Now, with this, we can also go through and look at gene mapping. In the case of gene mapping, is based on the number of recombinants from looking at individual alleles that are being passed down. We are able to determine how far away these specific alleles are from each other. And so again, if you have something like a 17% versus 8% versus 9%, it's showing us that VG and the B allele are actually further apart on this chromosome. There's 17 map units apart, where the VGCN is 8% recombinants again, so those are 8 map units apart, and the CN and then the B, in this case, we're talking eye color, right? They are 9 map units apart. And so again, based on the number of recombinants and percentages, we can map genes out. And so when you go through and take a look at Mendell, again, nothing that he did was necessarily wrong. It just didn't dive in as deep as some of the geneticists have found here in the past. So again, Mendel versus non-Mendelian traits, gene mapping, gene linking, again, making sure you guys have a good understanding of that.